Welcome everyone. My name is Shai Arkin. This is the NEXUS Israel Dealmakers breakout session number 3, the Hebrew University Innovation Ecosystem, Cutting Edge Research. We have two wonderful speakers and myself, Professor Hayouka and Professor Oded Shoseyov. And I think by showcasing the wonderful research that they have conducted, you can easily see the sorts of innovative ideas that the Hebrew University comes up with. So we're going to start with Professor Hayouka, then head on to Professor Shoseyov, and I will finish. And please ask your questions at the Q&A box after each session. So let me just start by introducing briefly Professor Hayouka. He can simply be classified as one of the rising stars of the Hebrew University. In the very short time that he's been with us, he was able to publish dozens upon dozens of top-notch scientific papers, to register close to 12 patents, to come up with novel agents against bacteria that are resistant to antibiotics, and in short, exactly what you would expect from a rising star to do. So, Professor Hayouka, the stage is yours. Please, go ahead.
Thank you very much. First, I would like to thank the organizer for inviting me to share our research here today. And actually, I will show you how in my lab, we are actually combining basic research and applied research. And I will show you two main strategies or projects in my lab. And I will show you how we are developing novel antimicrobial chemical tools to improve food safety, but not just foodborne pathogen, but also clinically isolated or even plant pathogenic bacteria in order to develop crop protecting agents. But today we will focus on two projects on how we can improve food safety and food shelf life. So we are working on several aspects, and we are starting from purifying water in order to have the ability, of course, for living and also for the field. We're also working on crop protection agents, food preservatives, and we are designing and developing bioactive packaging in order to extend food shelf life and to improve food safety. And in general, we would like to prevent all food loss that we can imagine. So I will show you two strategies that we're working on in my lab. First, I will show you how we develop novel membrane active antimicrobial peptide and how we generate bioactive surfaces by immobilizing these active agents on several types of surfaces. We also are really interested in protein-protein interaction and peptide-protein interaction. And I will show you how we designed a new food preservative to prevent fungal growth. And I will show you how we can use these approaches in order to design food preservative and extend food shelf life. So in order to protect food, we first need to find the active agents. And I was really fascinated by the world of antimicrobial peptides, that we all have them on our skin, and all the organism on Earth has them in order to protect themselves against viruses, fungi, bacteria. You can see here several examples of antimicrobial peptides that each one of them has a different structure, very nice structure and very colorful, but very different. And the sequences and the structures are totally different. So we were fascinated, how can we explain the activity of these agents that they don't contain any similarities in terms of sequences or structures? What we noticed that all of them has a positively charged in order to interact with the negatively charged bacterial membrane, and also contain a lot of hydrophobic, at least 50% of hydrophobic amino acid, in order to penetrate into the bacterial membrane and to interact with the lipid chains. And then they interrupt the bacterial membrane and kill the bacteria in early stages. So what we decided to do, instead of focusing on one and single sequence of this antimicrobial peptide, we decided to use a combinatorial approach. And instead of incorporating a single peptide, we decided to use the two main important amino acids, the hydrophobic and the cationic amino acid, in order to incorporate them on the peptide chains And we used solid phase peptide synthesis, the regular methodology, but instead of incorporating single amino acid in each coupling step, we decided to incorporate a mixture of two amino acids. One of them is the hydrophobic and the other is the cationic in order to possess the antimicrobial activity. We explored these active agents and we studied how the composition, which amino acid we used, the hydrophobic and cationic, how the chain length of the peptide, and of course the stereochemistry. We showed that the 20-mer peptide has the most active optimal activity. At the end of the synthesis, of course, we cleave the peptide from the solid support, and then we generate a mixture of peptide up to 1 million of different sequences. They all contain the same two amino acids with defined chain length, but the order of the amino acid is totally different from one to another. We showed by this approach that we can use these agents also to protect water and to purify water from bacterial contamination. We use them on the field, in the greenhouse, and we showed how we can use them to incorporate them in several food matrices in order to extend the shelf life, and we incorporate them for designing of bioactive packaging and surfaces. So today I will focus on two projects, and I will show you how we did it. And actually what we did at the beginning is to explore the antimicrobial activity. You can see here, this is the minimal inhibition concentration, which concentration is needed in order to prevent the growth of the bacteria. And we were really surprising to see that the activity is towards gram-negative and gram-positive, and also towards antibiotic resistance bacteria. This is clinically isolated bacteria, and they all possess a good activity in the low microgram per ml, at least 10, maximum 10 microgram per ml. This is a very low concentration. And we then wanted to explore how we can use this compound in order to degrade and eradicate mature biofilm. These structures that bacteria can grow and adhere on several surfaces and then to replicate, and they extract a very thick extracellular matrix that prevent antibiotic and other agents to penetrate. So we used our amphiphilic compound, and we showed that we can very easily degrade and eradicate mature biofilm. This is methicillin-resistant Staph aureus. It's a very clinically relevant bacteria that cause a huge contamination in hospitals and quite number of cases of death all over the world. We also, of course, really interested in learning how these compounds are active, and using atomic force microscopy, a topographic, we can see after we immobilized on the surface of the mica, the AFM structure, a lipid bilayer that mimicking the bacterial membrane. It's a synthetic membrane where we can see that the peptide can self-assemble to a pore, and that's what our assumption of how they interact with the bacterial membrane, causing this pore formation, very uniform pore forming, and cause the leakage of the bacteria and the bacterial death. So we showed that these peptides are active and have good antimicrobial and antibiofilm activity. It's very easy and cheap to synthesize because it's a combinatorial library, so we don't need to purify them. And our assumption is that this mixture of compound, it's a kind of a cocktail. It will be very challenging for the bacteria to develop resistance towards this cocktail of compound. So we are performing experimental evolution in the lab. And you can see here, in every day, we are exposing the bacteria for a fresh media containing our agent. And you can see that very easily in green, the bacteria develop resistance towards the rifampicin and known antibiotic. And for our compound, we could not detect any change upon this treatment. Of course, we continue it till 80 cycles, and we still could not see any resistance occurrence cases. The next step was to design a bioactive food contact surfaces. We used several types of surfaces like polystyrene, glass, and other type of compound, and we immobilized it, covalently attached it, our compound, to these surfaces. What we showed that by using several linkers, so we use several chemical technologies in order to have a very flexible linker and to preserve the antimicrobial activities that we preserve. And you can see here several types of bacteria. This is a gram-negative and gram-positive bacteria. This is also clinically isolated bacteria. And we observed very good antimicrobial activity and eradication of the bacterial cells. We explored the mode of action, and as you can see here, these are the beads. This is beads without the immobilization of our compound, so we can see very hardly the bacteria. There is no absorbance of the bacteria on the surface, but when we immobilize our compound, you can see that there are many bacteria on the beads, and after several minutes of incubation, most of the bacteria are being lysed and killed on the surface of the beads. We developed several water purification system in our lab, and then we decided with Strauss incubator, the kitchen, the food tech hub of the company Strauss, we established a new startup called Prevera. And this, our main idea in this startup is to protect water and food from harmful effect of microbial contamination. This is actually based on our studies in the lab, and we use these antimicrobial peptides in order to develop a bead that will be incorporated in a water system in order to prevent bacterial contamination. So this is the idea. By attracting and killing the bacteria, we will have a safe and healthy water. And this is the company. And of course, thank you for Gal, the CEO, and Liat, she is the lead chemist in the startup. And next, what I wanted to show you, another strategies that we are working in the lab. I really like this project because I'm really interested in learning protein-protein interaction and to design peptidic inhibitor in order to prevent a vital protein-protein interaction. And by that, we can prevent the growth of fungi or any other organism. So we have several rational and combinatorial screening to design peptidic inhibitors. And we have several hits for a very central protein in the pathway of the growth of fungi, many fungi. It's very preserved. And for that, we established a new startup company called Bountica in order to use and to prevent spoilage of food that's caused by fungi. As you can see here, very easily, strawberry, and I'm sure all of us showed it in our refrigerator when we purchased fresh strawberry. And of course, the loss is really high and the economic impact is very high. There are many product recall. And the idea of this project is to find and design peptidic inhibitor to prevent this pathway, to prevent several protein-protein interaction in the fungi. And then we can use it as a food preservative, as a safe and as a safe preservative. And of course, there is a wide range of applications that we are aiming to start. So it will be fresh fruit juice and also several type, of course, of fresh produce. And this is, of course, thanks for the team, Shai, Fabian, and Gal Maayan. She is an assistant professor in Tel Aviv University, is my collaborator. That's it. I would like to thank, of course, my team in the Hebrew University, and of course, to my collaborator and for the funding, and for you for listening. Thank you. If you will have any question, I will be more than happy to answer.
Thank you very much, Tzvika, for that amazing talk. If anyone has any questions, please post them at the Q&A box. We'll go people 1 or 2 more minutes. Okay, so we have a question from Michael. What level of funding is needed for these sorts of projects?
Okay, to establish the startup companies, you mean? I guess at the beginning, we raised around $1 million for the first 2 years. And I hope by the end of these 2 years, we will be able to raise more funding for another stage of progress in our research and the development of the compounds. I hope it answers your question.
Okay.
There is another one. Thank you.
Okay. From an anonymous attendee, are there any health risks associated with these items, like pesticides, etc.? Are these compounds pollutive?
Okay, thank you for that question. And the idea of our approach is to use natural amino acids that will be totally degrade in our gastric system and might be used even for a new building block for other proteins. We did not observe any cytotoxic or toxic effect of this compound. And of course, we need to explore it more deeply in the future. Okay,
if there are no more questions, why don't we all thank Professor Hayouka virtually again for that wonderful talk. And again, we look forward to hearing more about these companies, and I'm certain about more companies in the future. And now it is my pleasure to introduce Professor Oded Shoseyov. What can I say about Oded other than the fact that he's one of the most creative, successful, innovative, entrepreneurial scientists that I have been privileged to meet. And when I think about Oded, I think about the fact that there are three different kinds of ideas. There's the idea that you say, well, that's not a very good one. Fortunately, I didn't come up with that. Then there's the idea that you say, well, it's very clever, but given enough time and resources, I would probably have come up with that. And then there's the kind of ideas that Professor Shoseyov comes up with. These are ideas that you say to yourself, wow, not in a million years would I have ever thought about that. And Professor Shoseyov does this again and again and again. And that's why he won the Prime Minister's Prize for Most Innovative Scientist. And that's why we are all in for a treat. So Professor Shoseyov, the stage is yours.
Thank you very much. I'll try to do the full screen here. Let me just find it. I apologize for that. Okay. Again, thank you very much for giving me the opportunity to present to you some of our studies. The title of my talk is The Plant Age, Materials for the Future. But before I start, I would like to quote the former Minister of Oil of Saudi Arabia that said that the Stone Age did not end for lack of stone, and the Oil Age will end long before the world runs out of oil. And I think that is for a good reason, because biomaterials are really so much better. But we shouldn't criticize our scientists for not making really good synthetic material from oil for a simple reason. They didn't have enough time. 200 years of modern chemistry, or maybe even 300 years from the Industrial Revolution is not enough time to make really good materials, while nature had billions of years to develop some of the most amazing materials that we wish we had in our possession. Yet, our orthopedic surgeons, they really enjoy to introduce synthetic materials into our body. They screw them, they glue them, and I don't think that this is a great idea for a simple reason. Synthetic material fail. Just like this plastic fork that is not strong enough for its performance, but that's not the only reason, because many times synthetic material can be very, very strong. However, their biomechanical properties do not really fit the surrounding tissues, and that's not why it's not a good idea to introduce them into our body. But the real reason why it's not a good idea to introduce synthetic materials into our body is because in nature, things are done differently. In nature, nobody takes my head and screw it into my neck, or take my skin and glue it onto my body. In nature, every living organism, whether it's a plant or fish or human being, it's made of cells, and the cells contain DNA, and the DNA encodes for nanobio building blocks. They self-assemble into scaffolds on which cells are growing and proliferating, organizing into tissues. They organize into organs, and altogether, that's the organism, life. So we at the Hebrew University, about 16 years ago, decided to focus on probably the most important nanobiomaterial for us, for human being, and that's collagen. Now, why collagen? Because we don't have anything more than collagen in our body except for water. So it accounts for about 30% of our dry weight. So I'll give you a few examples. Bone is about 50% collagen. Our skin is about 70% collagen. Tendons and ligaments close to 100% collagen. So I always like to say that anyone who is in the business of replacement parts for human being would like to have collagen. But admittedly, 16 years ago, when we started our research, there were already in the market, in the clinic, more than 1,000 different medical implants that are made of collagen. You know, simple things like dermal fillers that you can see on the right-hand side, artificial meniscus, bone grafts, and even leaflets of heart valves, all made of collagen. Then you should ask me, why bother? You just said that all these collagen implants were already on the market. But the reason is the source. The source of all these collagen products up until recently was dead bodies, dead pigs, dead cows, and even human cadaver. And that's not a very safe source. And that's why already in 2007, the FDA made a notice asking all the companies and scientists to start to look for better and safer alternatives. So that's exactly what we did. We cloned all the 5 human genes from human, and by genetic engineering, introduced these genes that are responsible to make human collagen in our body into transgenic tobacco plant. So now the plant has all the information to enable it to produce human collagen in the plant leaves. The technology was matured, and today in Israel, a company that was established based on the technology has about 25,000 square meters of greenhouses all over Israel, from the upper Galilee in the middle of the country in the south. The farmers receive these small tobacco plantlets that looks exactly like regular tobacco, except contain 5 human genes that teach the plant how to make human collagen. We grow them for about 50 to 60 days, harvest the leaves. The leaves are transported by cooling trucks to the factory in the north, where we start the process. Now, if you ever made pesto, you know how to make collagen. Yes, big food processor, you crush it, get the juice, concentrated protein. And the concentrated protein is then transferred to Rehovot, to the science park, into clean rooms where we purify the collagen, and the end result is collagen, which is identical to what we have in our body, except it's brand new, and of course, much safer because plants do not harbor human pathogens. And from that collagen, we make different medical implants, 2 already completed clinical trials and reach the markets in Europe and Israel. But this is not the end of the story, because about 6 years ago, now we realized in the lab that plants are really able to make complex proteins like collagen, as I just told you. And we asked ourselves, what is the next target? What is another protein that we would like to produce in plants, something that will be useful for us, for human being? And we came to the conclusion that it must be antibodies, humanized antibodies. Now, why antibodies? Because antibodies are the guardians of our body. They protect us from infectious disease, cancer, etc. And they make excellent drugs, biological drugs. For example, Humira, which is a humanized anti-TNF alpha antibody, which is an excellent drug for rheumatoid arthritis, Crohn's disease. And it is a great drug, except one big problem: very, very expensive. The cost per patient is $35,000 per year. That's roughly $100 a day. So the end result is that only 1 out of 1,000 people in the world that would like to enjoy Humira can afford it. The question is, why is it so expensive? So some will say, well, maybe there are patents. Well, there are no patents anymore because they lapsed. Well, maybe there's no competition. There is competition. In fact, there are today 5 companies in the world, 2 Americans that produce Humira, but the price didn't drop significantly. Why? Because it's very expensive to produce. Unlike small molecules that can be produced chemically in reactors, humanized antibodies, biologic drugs, are produced by very sophisticated bioreactors in very delicate mammalian cells. And the systems are expensive. The media that you have to feed is expensive. The clean rooms, so all that is very, very expensive. So we asked ourselves, well, if it is so expensive, why produce there? So let's clone the genes of the antibody and stick it into a plant and grow it in greenhouse at a cost of lettuce or tomato. So that's exactly what we did. So what actually we did was we simply cloned the genes from this expensive system and introduced it into the plant and to the greenhouse. And the target is not $100 a day. Our target is to get Humira to the patients for 70 cents a day. And at that price, even a farmer in India that maybe makes only $8 a day, he can afford it. He doesn't even need the government. Well, trees are also quite amazing. I mean, look at the sequoia trees. They stand there at very low, withstand at very low temperatures, carry hundreds of tons for hundreds of years. And not just cold temperature, but also hot temperatures, UV light. And I tend to ask myself, how come that they are so resilient? In fact, I'm not familiar with any plastic chair that is made of synthetic materials that can survive such harsh conditions for such a long time. And so if you ask yourself the question, what is it made of? And you look by high resolution electron microscope into the heart of the tree, you find out that it's actually made of a nanofiber. That fiber is highly crystalline and very strong, and surprisingly is made of sugar. Well, it's not exactly the same sugar we drink in our tea. It's actually a polymer of glucose called cellulose, but it's not even a regular cellulose. It's in fact a highly crystalline cellulose, about 100 nanometer in length and about 5 nanometer in width. So strong that on a weight basis, it's about 10 times stronger than steel. So analysts all over the world today believe that this material, cellulose nanocrystals or CNC, is going to be one of the most important material for the entire industry. And not just transportation or construction, but in fact, even medicine, sports, food. They predict that in 2020 something, in fact, most materials and products on the market will contain some level of CNC. But here's the problem. Up until 7 years ago, there was no commercial source. So we, together with our partners in Sweden, developed a technology to cost-effectively produce nanocellulose. And today, in Israel, we have an industrial site to produce tons and tons of that material. Now, this is great, but the question is, okay, now when you have so much of that material, what can you do with that? Well, one fundamental property of these nanofibers is that when you suspend them in water at a certain concentration, which is about 2 to 3%, all of a sudden they make something that is called liquid crystals. And these liquid crystals, when we coat them over any substrate, it can be plastic or paper, wood or concrete, once the water evaporates, they self-assemble into an extremely strong film with excellent oxygen barrier properties. So why is it so interesting? because now all of a sudden, if you take this material and you add nanoparticles like zinc oxide nanoparticles, you can block both UV and oxygen. And that's what's really needed for packaging. In the old days, you see on the left-hand side, that's how we used to pack our food in tin, which is great, but very expensive and heavy. So we moved to flexible packaging, which is a great invention. Flexible packaging, in fact, is laminate of low-density polyethylene, and on top of it, some aluminum. So excellent product, huge industry, $1.5 trillion industry, except one big problem. Once you laminate aluminum on low-density polyethylene, there is practically no way back. So it pollutes our environment. So we have been able to develop, using that material, excellent packaging, which is totally biodegradable. And with that technology, we hope to get rid of plastic and aluminum from our environment. So what's for the future? I believe that we are on the verge of the plant age, because plants can produce everything, not just antibodies or collagen, or of course cellulose that they produce themselves anyway, but in fact, anything that nature created so far for us. And then by using technologies like 3D printing, we'll be able to take all these individual bio-building blocks and even print a heart. Now, this heart is not going to be as good as we can get from a donor. It will be better because it will be produced from brand new, much better proteins. Now, this is, of course, going to take a long time, but there's still already things that we start to print using these collagen and other biomaterials. And I would like to wrap my talk by quoting Pavlov, who said that if you want a new idea, you should open an old book. And I say that the book was written. In fact, it was written over billions of years of evolution. And the text is the DNA of life. We don't need to reinvent the wheel. All we need to do is read this text and start our progress from there. Thank you.
Well, thank you very much, Oded, for that amazing talk. I'm sure it has spurred some thoughts and questions in the audience, which they are encouraged to post in the Q&A box. Okay, you have a question. Where does marijuana fit in?
Well, this is another subject. In fact, marijuana or cannabis doesn't fit that particular, those particular aspects that we studied. We did made research at the university to develop a remote sensing of cannabinoids in cannabis, but naturally the time do not allow to talk about all these. As you may know, the Hebrew University actually established the first national cannabinoid research. So there is a lot of activity going on right now at the Hebrew University, and I encourage you to take a look at the website of the Center of Cannabinoid Research and study more about it.
Professor Shoseyov, you have another question. When do you see this reaching the market at affordable prices?
So that's a great question. As for, I would say, I can respond for the three technologies. So collagen is already there. Their products are ready for diabetic foot ulcer and another product for repair of tendons and ligaments, which are affordable, and they're already on the market. With respect to 3D printing of organs, I think that will take time, but I believe that the first products will be able to be on the market in 4 to 5 years. With respect to the Humira, the humanized antibody, we already signed an agreement with a European pharmaceutical company that and we hope to start clinical trials already in 2022. If everything goes well, 3 years from now, it will be on the market.
You have a question. Do you try to interest global students with your ideas?
Absolutely. In fact, in my lab, currently about 50% of the students are foreign students from China, from India, from the US, from the UK, and of course Israelis. So the answer is definitely yes.
Another question from an anonymous attendee. When could the packaging be brought to the market on large scale?
So that's really on its way. One of the partners of, or the shareholders of Melodia is Klabin, which is a very large forestry pulp and paper company from Brazil. And then they intend to launch the first packaging product within a year from now. We have discussions with other major global companies that are now testing these prototypes. And I believe that in less than a year from now, we will start to see these products on the market.
Do you see interest from the packaging industry in CNC?
Absolutely, huge interest, mainly because the European regulations, as I'm sure all of you heard, Nestle, for example, announced that in 2025, they will have absolutely no plastic packaging or synthetic packaging in their products. And to make that claim and get there, they will need these technologies. So I believe that that we will see that very soon.
Okay, let's move on. I have one last question, but that's for the moderator. Why did you put me in the same session after Oded? He's an impossible act to follow. So really, thank you very much. It's an amazing, amazing piece of work. So last and hopefully not least is yours truly. Okay, where am I here? Okay, so I want to talk about a small company that I started at the Hebrew University just this last November. And if you ask yourself, why did it take me so long after 2 decades to start a company? Well, up until December 2019, if you sat in a cafe in Hamburg or in London or in New York or in Tel Aviv or in Tokyo, you really were not that frightened or interested about viruses. You thought that dengue is something of the Southeast Asia. You heard of Zika, that's somewhere in South America, etc. But really, viruses were not a major concern to you. Well, I think we all realized that that was slightly wrong. And today we recognize the problem. We recognize that viral diseases are global. We can no longer rest on our laurels and be certain that if we have a viral issue in one part of the world due to globalization, it will not head to our doorstep and cause a shutdown of practically life as we know it. So a simple question, what are we doing to prevent the next pandemic? Not COVID-19, but rather COVID 2021, 2022, etc. And I'm going to give you a dire outlook. So the World Health Organization recognized 16 pathogens with these frightening names as posing the greatest risk to mankind. So some of them we are familiar with, COVID-19, obviously on the bottom left, things such as Ebola, Zika, chikungunya, MERS, SARS, and many other frightening organisms. So these are recognized by the premier body that is responsible for the world's health, hence called the World Health Organization, as posing the greatest risk to us. And what is the pharmaceutical industry doing in return? The answer is it's only studying 6 out of these 16 organisms, 6 out of these 16 pathogens. And in fact, if I were to give you this slide prior to December of 2019, obviously there was not a single pharmaceutical company that was studied a coronavirus because it was not deemed as a threat. And as you can see, there are numerous very dangerous organisms which are currently not under research and development in the pharmaceutical industry. So how do you treat viral diseases? Why is it so complicated? Why are we not studying all of these organisms? So there are 3 major avenues to treat viral diseases. Obviously, there's vaccination. We've all heard about that. Israel has been particularly successful using the Pfizer vaccine. There's plasma convalescent therapy. This is basically using somebody else's antibodies. It could be from an actual person that got well, or it was an antibody was engineered, such as the sort of things that Oded mentioned. And finally, there are antiviral drugs. You would say, wait a minute, I've heard that vaccination is so successful. Well, it wasn't vaccinations that have kept Magic Johnson alive since the 1990s. It was antiviral drugs, because for some viruses, despite our best efforts, we were not able to generate appropriate immunizations. Hepatitis C is another very, very famous example. So that's what we're trying to do. And here's another problem. So to date, unfortunately, there are no validated platforms. There's no standard route to generate antiviral compounds. So it's normally a one-shot, specific research focused on one particular compound to develop, but we don't have a routine procedure to churn out new antiviral drugs. And even if we had such a method, the problem is that viruses change rapidly. We've all heard about new mutants of SARS-CoV-2, the causative agent of COVID-19, and our fear that they would be resistant to the new immunizations. The same would also hold for any antiviral agents that we have. So the conclusion is that we have developed a procedure in my group in the last 2 decades. And now, because there is an urgent need to develop new antiviral compounds, we formed a company called ViralBlock that is able to generate antiviral compounds against most human viruses quickly and efficiently. And we are also able to map the resistance potential against this. So we can have one up to preempt the ability of the virus to develop resistance. Okay, so if you want to develop an antiviral drug, you need to have a target. Okay, if you want to disable an automobile, you might want to choose, okay, I'm going to take out the carburetor, I'm going to puncture the tires, I'm going to, I don't know, destroy the transmission. There's a slew of things that you can do, and viruses are not terribly complex. They have a dozen or so components. Which of them do you want to target in order to inhibit the virus? And what we have chosen, you can see a sketch of the flu virus on the left and SARS-CoV-2, the causative agent of COVID-19 on the right. We have chosen a target that most have ignored. It's these tiny little things. These are ion channels that viruses have. Okay, these are small proteins, small machines that are located in the membrane of the virus. And what do? Well, what do ion channels do? Ion channels enable salts to pass through the membrane. Every organism has a membrane that encases him, and viruses have them as well. Most viruses, at least, most human pathogens. But the membrane is a wonderful casing, but the problem is it does not enable the organism to control its internal salinity and acidity. And that's why they need special proteins to enable them to control the passage of salt in or out. Now, so far you would say, well, that sounds like a very interesting, esoteric, basic scientific issue, except that manipulating channels is very, very common in medicine. If you just look at the list of compounds on the bottom, who of us has not used lidocaine when they went to the dentist? What does lidocaine do? It blocks a sodium channel in our body. Xanax, what does Xanax do? It blocks a particular channel in our brain. And all of these very common compounds block and manipulate the activities of channels. So if we can manipulate channels and viruses, what we will be able to do is to inhibit them. And that's precisely what we want to do. So what is the task at hand? The first thing that we need to do is we need to identify the particular channel in the virus that we want to inhibit. Then we have a specific assay that we can screen for blockers. We identify inhibitors, and then we have a third procedure in which we assess the resistance potential, but I'm not going to go into that today. So how do we do all of this? We do all of this in a wonderful laboratory. We do all of this in the laboratory of a bacteria. So we don't do this in a test tube. We actually express the viral channel in bacteria, and the viral channel impacts the bacteria. And then all we need to see is, can we change the behavior of the bacteria? And that will be indicative that we have inhibited the viral channel. Using bacteria for this enables this process to be very rapid, enables us to change mutations and screen new channels within a week. So this is a very rapid and cost-effective procedure. So we have 3 independent assays. How do we test these? It's quite simple. In the assay on the left, it's an assay in which the viral channel harms the bacteria. So when it's active, the poor bacteria don't grow properly. Therefore, you can easily identify a blocker because the blocker alleviates the detrimental impact of the channel and allows the bacteria to grow. So all we need to do is to monitor growth. And the assay on the center, it's exactly the opposite. Here we have strange bacteria that have a particular defect that is able to be fixed by the viral channel. So in this instance, the bacteria cannot grow unless the channel is active. Therefore, a blocker would retard bacterial growth because it removes the positive benefit. And we have a final assay in which the viral channel changes the bacterial acidity, and therefore a blocker will negate that change. So just to show you what this actually looks like, here are Petri dishes where you can grow bacteria. This is the first assay. You have happily growing bacteria in the red Petri dish on the top left. When you express the viral channel, it pokes lots of holes in the bacterial membrane, and the bacteria die. And then when you give the bacteria an antiviral drug, believe it or not, this antiviral drug is not only beneficial to humans, it's also beneficial to the bacteria because they're suffering from precisely the same problems that we are, okay? We have basically challenged the bacteria with the same virus that causes us problems, and we are relying on the resilience and the bacterial ability to avoid threats in order to look at this assay. The second assay, as I mentioned, is the opposite. Here we have a bacteria with a defect that you can see on the top left do not grow. When you grow when you express the viral protein there, suddenly they are able to grow. And therefore, when you add something that negates the positive impact of the viral channel, the bacteria are dead again. The final assay is a colorful one, because here what you do is you monitor the acidity with a particular pH indicator. I'm sure you've all done these experiments in high school in some age. So you have bacteria that have a certain color because of a certain acidity. The viral channel enables salt to enter, and therefore the acidity will change and the color of that bacteria changes. All you need to do is to add different chemicals from a chemical library and look at which bacteria does not change or changes to a different color. And that will indicate that you have once again inhibited the viral protein. So where do we stand on COVID-19? As I told you, the first thing that we need to do is to identify channels. And we've done exactly that. We've identified 2 channels in the virus. They're called the E protein and 3A protein, and we have confirmed their activity. So this is great. Now we have a target in the virus that we can inhibit. We have something to latch on. We've screened 3,000 chemicals that are approved for human use. This procedure is commonly called drug repurposing. So we have tested 3,000 chemicals in each of the 3 assays that I've mentioned to look for chemicals that inhibit the E protein or the 3A protein. And fortunately, we were able to find inhibitors for both of those proteins, and in appropriate biosafety level 3 facility, we were even able to show that many of them have antiviral activity. So this is actually a confirmation of our entire scheme. We were able to inhibit channels with particular compounds, and then we show that these compounds inhibit the whole virus. So here are the results. So all of these compounds, you can see the names underneath. We've obviously patented all of them. These are all compounds that are approved for human use, for other indications, obviously, because there are no compounds except remdesivir. And again, it's not nearly certain if it is actually efficacious or not, but all of these compounds were developed against other indications. And you can see, for example, 5-azacitidine is able to inhibit viral replication close to 98%, peroxifor above 50%, and further on. So as you can see, this entire flow, this platform is quite advantageous. So what is the next steps? In the near term, we are looking at conducting phase 1 or phase 2A trials on the generic drugs that we've identified against COVID-19. This obviously costs a fair bit of money. So this will necessitate raising additional funds because the company is only funded to conduct the basic research and identify channel blockers. And in the medium term, we also want to identify repurpose inhibitors for other viruses such as dengue fever, West Nile virus, Zika, and many, many more. So as a quick summary, we are a very, very new startup, only started at late 2020 due to the rampant pandemic. We are looking for antiviral compounds using proprietary approach, both cost efficient and rapid. We have several disease targets. And in fact, we've already finished screening 1 channel in dengue fever as well. And we will target, as is written here, COVID-19, MERS, dengue, Zika, West Nile, and many more. And our current assays are already a slew of chemicals that inhibit each of these compounds, which are approved for human use. And our target is to take it to the clinic and actually test it on patients. So hopefully that was sufficiently swift. I'll be very happy to answer any easy questions that the audience have. And if you feel timid to do so at this point, feel free to contact me at the email that is registered here. Okay, so I have one question from the audience. How scalable is the method of experimentation? How long does it take to identify an inhibitor for a new virus? It takes us about 2 months. So the great thing is that for us, a channel is a channel is a channel. We've already cloned channels from lots of different viruses, frightening ones such as Ebola, and less frightening ones such as flu, etc. So give us a virus and we will clone a channel, characterize it, and identify inhibitors within a period of 2 months.
Can I ask a question?
An easy one always, Oded.
So actually that brings me back to the cannabis. Did you test channel blocking agents coming from natural sources like cannabis oil?
We used a library of something like close to 3,000 approved for human use drugs, but I think it's a wonderful idea to look at other natural compounds because they might be easier to regulate. So you're in Rehovot, I'm in Jerusalem, it's 20 minutes away. Let's work together on this a bit.
Absolutely, we have a deal.
I have another question. What are the relative costs of the produced medicine? So all of the compounds that we are looking for are approved for human use. I think even if I were to identify a brand new compound today that is tremendously efficacious, none of us will be able to see it in the clinic in the near future. that's why we decided to both limit the chemical space by focusing on drugs that are approved for human use. So the cost for the specific medication depends on the particular medicine that we have found. So, but they're not substantial. Okay, well, if there's no further questions from the audience, I'd like to thank the first two speakers coming from the Faculty of Agriculture, I forgot to say, which is a powerhouse of wonderful science and ingenuity and entrepreneurship. And you saw two great examples. And I hope that you all are enjoying everything that this wonderful conference has to offer. And have a great continue of NEXUS Israel. Thank you very much for attending.
Thank you.